Okay, hello everyone. My name is MacKenna Tamanaha.
And my name is Clara Lee, and over the summer, we researched the bacterial levels in Kahaluu area for our K Fellowship project.
We would like to express our gratitude to Papa Jack for this research opportunity. We were able to learn a lot from this summer's research, which wouldn't have been possible without the impact he left at Iolani.
So Kahaluu is the main location for our testing. For those who don't know, Kahaluu is located on the windward side of our island, just north of Kaneohe. We chose this location because of its reports of infections and high bacteria levels.
So this is a compilation of a few older news clips from when Kahaluu locals started to get these bacterial infections in 2014. Sorry.
During one of our summer meetings, the residents came to report that the canoe paddlers were getting cut heels or open sores and infections.
I'm staying out right now, but I've been going in the water all the time.
Junior used to fish in these waters every day and now knows why a simple scrape just wouldn't heal over the past 10 months.
The doctor said it was called MRSA.
For Lenny Soares, who doesn't want his face shown, a cut he got fishing in the windward waters turned into a life-threatening emergency just a few weeks ago.
My leg started to swell up like a balloon. So it was like 3 times the size.
And this was a big motivation for us to pick Kahaluu as our research site because of its existing presence of pathogens, which are bacteria that can infect the human body.
During one of our summer meetings, the residents
After MRSA, bacterial, and other antibiotic-resistant infections were reported from Kahaluu area, the DOH began to investigate the cases of these infections. And then they found high levels of Enterococcus in the beach, which is a fecal indicator. This means that the contamination is caused by feces in the water, assumed to be from either nearby pigs or cesspool overflow or leakage.
So cesspools are tanks in the ground filled with sewage waste. There are currently around 88,000 cesspools in Hawaii, 43,000 of which are current threats to water resources. They discharge over 53 million gallons of untreated sewage into the ground each day. This sewage leaks into groundwater, which eventually flows downstream to contaminate lagoons and bays. In untreated sewage tanks, there are disease-causing pathogens, which are serious health risk when paired with antibiotics from household use.
When antibiotics and pathogens inhabit the same space, the perfect environment for antibiotic resistance is created. Antibiotics end up killing the susceptible bacteria, but leave few resistant ones because they're unable to kill them. This leads to those antibiotic-resistant bacteria becoming the majority in the waters and transferring their antibiotic-resistant genes to other bacteria. This is most likely what's happening in Kahaluu waters, and this is especially dangerous to locals because antibiotic-resistant infections are difficult to treat and are lethal. In fact, they are one of the leading causes of morbidity and mortality in the world.
These issues urged us to research more into these topics, which leads us to our research questions of how do the levels of Enterococcus in the Kahaluu lagoon, stream, and beach compare, as well as how do antibiotic-resistant bacteria relate to the levels of Enterococcus and total coliforms in the Kahaluu waters.
We chose to sample 3 different locations because we hypothesized that the contamination came from cesspools, and therefore bacterial levels would be more concentrated in areas closer to the discharge and more dilute in areas farther away. So we sampled the stream up in the neighborhoods closer to the houses' cesspools, the beach by the dock, which would be the furthest away from the leakage, and the lagoon, which is in between the two. We only chose 3 locations to keep sampling and testing as simple as possible while generating a diverse enough results for comparison.
Our first goal was to quantify the levels of Enterococcus in each location to determine which area was the most dangerous. We chose Enterococcus because it may be a fecal indicator in Hawaii, and therefore it gives us more insight into the amount of bacteria in that water and the possible presence of disease-causing bacteria, viruses, and protozoa. The most important aspect of this goal is determining which location has the most amount of bacteria so that we can warn the community about it, because unfortunately on our trips to sample sites, we've seen a boy and his dogs playing in the stream, people fishing from the lagoon, and boaters and paddlers entering the ocean. So we want them to be informed about the health risks of entering these waters.
We ran call alert tests in order to quantify the levels of Enterococcus in our water samples to their respective location. We had to adjust the concentrations of the kits from a very concentrated ratio of 1 to 10 in terms of sample to water, to a ratio of 1 to 100, which was a little too concentrated, to a ratio of 125 that was just right, so that there was at least one clear well, not too concentrated enough to change the color, ensuring that our results were not inflated and able to be quantified. 3 sets of tests yielded genuine results over the summer. Those 3 sets of tests were quantified by counting the number of yellow big wells to yellow small wells to find the most probable number of Enterococcus levels through an IDEX table.
This is a bar graph of the average Enterococcus colony forming units per 100 milliliters of water in each location from our 3 test runs. We also have box graphs of each location's 3 test runs on the side. All of our graphs show the same trend, that the lagoon has the most Enterococcus levels, and then the stream, and lastly the beach. This answers our first research question, showing that the lagoon poses the biggest health risks to locals, even though all locations exceed DOH safety standards for Enterococcus in waters.
Our second goal was to grow bacteria from our water samples and identify pathogens among them, and then test those pathogens for antibiotic resistance to get a better understanding of the infectious bacteria in Kahului.
In order to decide which antibiotics we were going to test for susceptibility or resistance, we first had to understand the antibacterial environment in wastewaters, especially in the Kahului area. From a research paper of a pilot study on a cesspit tank in India, we learned about antibiotic-resistant in cesspools. And from a local paper on Hawaii contaminants of emerging concerns, we learned about the presence of different antibiotics in Oahu's waters. These factors led us to choose 6 antibiotics: ciprofloxacin, erythromycin, gentamicin, penicillin, tetracycline, and vancomycin.
To grow bacterial colonies, we spread different amounts of water samples directly onto marine agar. We then picked single colonies and streaked them onto another marine agar plate to isolate bacterial species for DNA extraction.
The first test that we ran were PCR tests, which sequences the 16S RNA gene to identify the species of the bacteria. This was done in 2 parts: the InstaGene and PCR testing to isolate DNA or to extract DNA and replicate the 16S gene, and the gel electrophoresis to visualize DNA before sending it off for sequencing. We sent off 11 samples and 45% of them were pathogenic. In the beach, Schuanella coralli; in the lagoon, Schuanella coralli and Enterovibrio nigricans; in the stream, Pectobacterium, E. coli strain CY598, and Pentawea stewarti subspecies indologenes.
We next used the Kirby-Bauer method to test these pathogens for antibiotic resistance along with other bacteria. We used colonies from streaked plates of the identified species and inoculated it in Mueller-Hinton broth to grow up a concentrated enough sample to plate a lawn of bacteria on a new agar to be sampled with antibiotic discs.
We let those stamped bacteria lawns grow in the incubator overnight. Then we measured the zone of inhibition, which is a circular area around the disc where the bacteria was killed by the antibiotic and couldn't grow. On the rightmost plate, you can see that the zones of inhibition are all present and large, meaning that the bacteria is susceptible to all 6 antibiotics. However, in the zoomed in parts of the other plates, there are small or no zones of inhibition, meaning that they are resistant to those antibiotics. These pictures are color-coded, so the blue pictures show penicillin resistance, the pink pictures show vancomycin resistance, and the yellow picture shows erythromycin resistance. Our findings revealed that 60% of the 5 pathogens tested displayed antibiotic resistance. The only drawback in these results is that the Mueller-Hinton broth didn't grow any saltwater bacteria to the concentration it needed to be. So we don't have enough data to compare or correlate Enterococcus and antibiotic resistance in the Kahului area.
However, we managed to collect data despite these obstacles. Our IDEX kits provided the levels of Enterococcus in our sampling sites, all of which exceeded DOH safety standards. And we performed PCR tests in the Kirby-Bauer method to prove that there is a presence of antibiotic-resistant pathogens in all 3 tested Kahaluu locations.
From our struggles and failures, we came up with a few possible extensions to our project that will yield more diverse data and which would help us answer our second research question and understand the bacterial contamination in the Kahaluu area. Our first suggestion for future research is expanding sample size by either testing sites in these heavy cesspools areas or sampling the 3 test sites that we did more often.
Our second suggestion is to test different broths for better results with the Kirby-Bauer method, because as mentioned earlier, the Mueller-Hinton broth seem to grow freshwater bacteria and not saltwater ones. So experimenting with marine broth for saltwater pathogens would be a good option for testing antibiotic susceptibility in saltwater bacteria.
Our third suggestion is to measure the levels of antibiotics in the waters to look for a correlation between antibiotic levels and antibiotic resistance. We originally planned to do this with the help of our mentor, Dr. Dulai, at her lab at UH Manoa, but unfortunately, we didn't have enough time to. I plan to try and finish this goal along with other suggestions over the span of the school year as I continue this project in independent research.
Throughout our experiment, we've had to overcome many obstacles. One of the main struggles in our project was finding the right ratio of water sample to sterile saline so that the bacterial growth would be just right on our plates. Our trips to Kahaluu were often several weeks apart, and bacterial presence fluctuated, so it was often a trial and error process. We learned a lot from this summer, especially in regards to the research project, and we would like to share some advice for future K fellows or research students. First off, don't be afraid to fail. Mistakes are made, and so many don't end up working out, but you always learn from your mistakes and improve your work because of it. Secondly, expect so many things to get in the way of your research, such as scheduling conflicts, topic issues, and flat-out mental blocks. But no matter what, you'll overcome them with time. Lastly, stay productive as the work will pile on if you don't. And don't be observant, and be observant by taking notes because that will help when it comes to writing your paper and their presentation.
Lastly, we want to give a big thank you to our mentors, Dr. Chan and Dr. Dulai. Dr. Dulai helped us out with expert input on cesspool leakage and its relation to antibiotic-resistant bacteria, and gave us guidance on testing sites in Kahaluu. And Dr. Chan helped us through the research process and helped us learn a bunch of protocols for our tests in the lab.
Thank you to Mr. Tong for being a big help over the summer. He supervised us and was a very useful extra set of hands that helped us out with our work in the lab when Dr. Chan was working with us virtually.
Thank you to our families who supported us at Iolani and throughout our research process, especially giving us rides out to Kahaluu to do our sampling.
Last but not least, thank you to all the wonderful friends who also supported us and helped ease our anxieties about this project.
And that's all we have for you guys. Thank you so much for coming and listening.